Raptors were recently at the University of Johannesburg's Doornfontein campus to visit the Laser Research Centre and to find out more about their groundbreaking work in the field of laser applications. Here's what we learned.
We started the centre approximately in 2004, and we were approached by the National Laser Centre to conduct a feasibility study on the use of lasers in medicine. We started the work in wound healing and specifically in diabetic wound healing, as this utilises lasers with low power densities, and it's very underutilised and unknown in South Africa, despite the fact that it's cheap, they're transportable, and they can be utilised for a variety of different disease conditions. So ideally, we would like to promote phototherapy, especially in South Africa, and we would most certainly like to see that phototherapy be utilised as a complementary medicine, not only in cities and in big medical institutions, but in the rural areas, because it is actually a very cheap treatment modality.
So our work is done in the laboratory on tissue culture. So we grow fibroblast cells artificially in a petri dish or a cell culture dish in the lab, and then we will stimulate a diabetic condition, wound the cells, and then expose them to the laser, and then follow certain biochemical pathways. The lasers that we use are basically diode lasers. So lasers are classified according to what medium is being activated within the laser to basically give off the light. So we work with diode lasers, it's got a dye inside it. And then the lasers we use are very low powered, roughly about 100 milliwatts. We use different wavelengths, typically in the visible red to the near infrared range, and that's because the light has different penetration depths. So the higher the wavelength, the deeper the laser will penetrate into the tissue. So our laboratory is a state-of-the-art laboratory. We've got the tissue culture facilities where we will grow our cells artificially. And then to study the biochemical tests or the downstream effects of the light on the cells, we have equipment like a flow cytometer, which can count exact number of cells, for example, dead cells or live cells. We've got a PCR machine, which looks at the gene transcription or the expression of certain genes within cells. We have a live imaging microscope where we can actually grow cells within the microscope. The microscope has a little incubator around the stage, so within that chamber we can grow our cells, and we can visualize them and take videos and photos while the cells are actually growing, instead of just taking the endpoint photo.
The University of Johannesburg recently received a major boost to its fast-growing research and innovation efforts with the announcement of 5 new research chairs through the South African Research Chairs Initiative, SACHI. And one of them was awarded to Professor Heidi Argumse in the Faculty of Health Sciences for laser applications in health.